afternoon. We are team number
7.
7, conformed by Marco Barreto and Gary Lara, and we are talking about our project, which is about water pollutant agents. First, we're focusing on the Zapotlan Lake, which is located in the municipality of Zapotlan el Grande in Jalisco. We can see it here in red in the image. It's the second largest lake, only behind the Chapala Lake one, the blue. Well, these lakes, as big water bodies, are constantly monitored by the Jalisco State Water Commission, or SEA, Comisión Estatal de Agua, at 4 sampling points in the lake of Zapotlan. Um, these monitoring take information about water quality variables, which are those ones that allows us to measure the state of a water body and if it is safe or health for the people or the biodiversity to stay there or around there, that water body. Some examples are, total suspended solids, chemical oxygen demand, biological oxygen demand, nitrogen and phosphorus content, acidity or pH, temperature, among others. These are often measured in laboratories or authorized facilities from samples of water taken directly from the water body. For example, here in the image, we can see 2 inspectors taking a sample from a river in a 1-liter bottle, for example. Well, for our project, the data was extracted from the SEA Commission. This information is public, is on their website, and as a time series, this database was downloaded as a time series with monthly periodicity from September 29, 2009 to April 2018. This dataset includes information of 30 all continuous water quality variables. And the purpose of this, or the purpose of obtaining this dataset was to analyze it exceeds the permitted levels in order to associate it with different activities carried out around the lake. For example, the agricultural activity, the industrial activity, and also to estimate the potential adverse health effects in the populations that are near, particularly the Zapotlan el Grande population.
So in here, we have the original data set with the 30 variables, which they're the variable, the abbreviation, and the units. And there are 30 variables in here, but not all of them are useful for us, since it contains a lot of data from them. So what we did from here is that for our purposes, we only selected 4 variables, which are the coliforms, the total solids, the nitrogen, and the biochemical oxygen demand, which help us because they are the most commonly related with the general adverse effects on the environment, flora and fauna of the lake, and potential negative effects on the health of population around the lake. Next.
Well, as a brief explanation of these 4 variables, the total solid is the amount of solid mass per liter of water. With solids, we are talking about the matters such as dirt that can dissolve in the water. We are excluding from this all the objects or products such as bottles, tires, among others. Why this is variables that it's measured? It's because it's related with the sludge deposition, contaminant absorption by these solids, and also pathogens protection, among others. The total coliforms are the amount of coliform bacteria. Coliforms is a type of or species or group of bacteria which are encountered or principally founded in human and animal feces and are related in case of a person to consume this water with a high concentration of total coliforms are related with some diarrheal diseases and even intoxications. The total nitrogen and phosphorus, well, these are related with a phenomena called eutrophication, which is the overpopulation of algae species in a water body. Since nitrogen, we are focusing in nitrogen in this study, in this project, and since nitrogen is a nutrient for the plants, if there is a high concentration of nitrogen in the lake, then the algae species and the plants will start to grow and overpopulate the lake, and this will also conduce or cause increase of uptake of nutrients from the algae, and then the other species will be a lack of nutrients for those species. And then the last, which is biochemical oxygen demand, which is the amount of oxygen required to stabilize the matter, which is, and this is related with the oxygen availability for the animals that live in a lake. For example, if there is a high biochemical oxygen demand, this means that the matter that is in the lake, non-living matter, will require a lot of oxygen. Then the animals, for example, the fish that require also oxygen, they don't have enough for live and they will start to die.
So from the previous information, we present our 5 different scientific hypotheses, which are the number 1 is the lake location. There are 4 different sampling points from where the water is taken, and this has an effect over the biochemical oxygen demand variable. The number 2 is the season. We have autumn, winter, summer, and spring. And these different seasons have an effect over the nitrogen concentration levels. The number 3, which is the time and over the years, has an effect over the biochemical oxygen demand variable, which can be increasing or decreasing depending over the years. The 4th hypothesis is that agricultural activities such as farms or planting crops around the lake have an effect on the total solids variable, meaning that it might increase or decrease. And the 5th one, we have an introduction of sewage treatment plants around the lake have an effect over the total coliform concentration, meaning that these treatment plants might help to decrease this concentration, or they don't work at all, and the coliform concentration might be increasing over the time. On to the next slide. We have that for our first scientific hypothesis, which is the lake location and relation with the oxygen demand. We have that X represents the oxygen, and its subscript represents the station. So we have 4 different stations that the samples are taken from. So mu1 represents the mean of x1. So our statistical hypothesis tells us that each different sampling point should be equal. But our alternative hypothesis tells us that the variable from each sampling point might be different. Coming to the next slide, we have our second scientific hypothesis, which is the season, having autumn, winter, summer, and spring, and relation with the nitrogen concentration levels. In this time, we have that X represents the measurement for total nitrogen in that season, being x1 spring, x2 summer, x3 autumn, x4 winter. And mu1 represents the mean of x1 and so on. So this time we have that our statistical hypothesis tell us that we check for an equality in between seasons. If there's not an equality in seasons, we can proceed to check if there's a major pollutant concentration in between seasons, and then we can see if the difference in between seasons and the concentration levels of the pollutants. Thanks. The next slide.
Well, for the third scientific hypothesis, which is the time over the years has an effect on the biochemical oxygen demand variable, well, we are seeking to perform a polynomial regression to see how it behaves for these 9 years of period of data that we have. For this polynomial of n order will be constructed in the form presented. And the hypothesis, a statistical hypothesis related to this polynomial will be that if the n term is equal to 0, this is the new hypothesis. That means that the polynomial does not fit well for the given data. And the alternative hypothesis is that this n term it's not equal to 0, so the polynomial fit well for the given data. For the fourth scientific hypothesis, which is the agricultural activities have an effect on the total solid variables, well, we are going to divide the data or the response variable total solids in two groups. The one from 2009 to 2013, and the second from 2014 to 2018. Why? Because in 2013, the berries productivity increases a lot. Well, in the previous year, but in that year, it was like notable. And then we are going to see if that agricultural change in the activities has an effect over the total solids variable. So there are x1 and x2. We took the means and our new hypothesis that there is no change between this, and the alternative is that there is a change between the total solids variables before and after this agricultural activity change. The fifth scientific hypothesis, which is the introduction of sewage treatment plants around the lake, have an effect over the total coliforms concentration. Well, the sewage water is the one that it is derived from the public use, which is, for example, the bathroom water, all the service water of the industries, among others. And then we have also, like the previous hypothesis, split the data in two, from 2019 to 2014, and then from 2015 to 2018. And we took we took the mean of each one. Why this periodicity? Because in 2014, at the end of this year, the Zapotlán El Grande municipality introduced 2 treatment plants. So we are going to see with this hypothesis if there is a difference in these total coliforms concentrations, which are encountered in this sewage water before and after the treatment plants were introduced. And also we want to compare the actual total coliforms concentration, or the most recent total coliforms concentration of the lake, with some upper limit standard published international.
So additionally, speaking of the statistical hypothesis, even though that some of the hypotheses are tests might seem the same, they represent different tests. for example, in here, the coliform concentration, we are trying to see the coliform concentration in this one. But some other tests, like the previous ones mentioned, we are trying to measure some different forms in this one. For example, the variation in between a period from 2009 to 2013 and a period from 2014 to 2018. So they represent different measurements and different tests. And for the plan, we have our plan for this project for the point estimation is to determine the mean and the standard deviation of the selected variables in order to the hypothesis tests. Then we are going to estimate the beta coefficients of the polynomial function presented in the third scientific hypothesis. And for the interval estimation, we are going to estimate the nitrogen concentration range or interval in several seasons. Also, we are going to estimate the confidence intervals of the polynomial function developed.
And last, for the hypothesis test, we will compare the means of the response variables, all the statistical hypotheses that we present before in different conditions, even though, as my partner Marco said, the statistical hypothesis seems to be equal. They are designed to prove different things. For example, the sewage water treatment or the agricultural activities change. Well, that is there are plenty of tests. And a second type of test will be to determine if the end term of our polynomial model is significant, the one presented in the third scientific hypothesis. Last, for the goodness of fit, we will perform the Shapiro-Wilk test to evaluate if our data follows a normal distribution. And then we will approximate a goodness of fit for a polynomial model by any method. Could be, for example, the Pearson chi-square test. Well, thank you for your attention, and that's all from our part.